Hey guys, very good evening, and I'm sorry I just took a little bit of time because I was going to do a live streaming. But anyways, we are here now, and let's get back to where we stopped the last class. Okay, so just give me a second. Before I start the session, so I have few questions to ask you guys. It's obvious that we have to do a technical check. So please tell me if everything is all right. You can see the video, the slide, the audio is clear, so we can continue on. Okay, please confirm. I think the poll, yeah, perfect, guys. Thank you very much. I think this should be enough. Yeah, all right. Yeah, perfect. So just give me a second. Just give me a second. Yeah. So in the last class, we were studying about the cell cycle, and we discussed some of the very interesting facts about the cell cycle and how the cell is such a programmed device like your mobile phone. And we understood what are the different important phases of the cell cycle, right? So if you try to memorize that in the cell cycle, we understood that the cell cycle can be divided into three important phases. That is the G1, the S phase, the G2, and then you have your M phase and probably the G0 phase. Okay. So now my question is, if a cell is not dividing, okay, let's say that the cell is not dividing, what are the stages that it will undergo? Can you please tell me in your chat box? So if a cell, I will write it here. If a non-dividing cell, I will have, okay, non-dividing cell. See what happens is you have a cell, okay, and the cell has gone under division and it has given rise to two daughter cells, okay. Now these two daughter cells will decide that I should not divide any further. Now tell me, in its cell cycle, what are the two stages it undergoes? So this is my question, guys. So let's go. If you want me to repeat, you can put it in the chat box. I can do that for you guys. Let me type the question. If a cell is not dividing, okay, what stages it undergoes? So this is my question, guys. Come on, let's go. Anybody with an answer? So chalo, as you answer, I will just move on with the subject. Okay, I will wait for your answer. So generally, in a dividing cell, in any cell that undergoes division, we know that the cell has to, undergo certain phases during the cell cycle, and we can divide this cell cycle into G1, S phase, and G2 and M phase. Okay, so we understood that in G1 phase, the moment the cell is born, the cell has to prepare. Basically, it has a growth phase. And G1 is also called as the gap one phase, or is also called as the growth one phase, as we have studied in the earlier classes. So here, many of the organelles also develop along with the metabolic activity. So the metabolic activity of the cell is very, very active. That's what we understood. And then it prepares for the DNA machinery. Okay, so the cell will enter into G0 phase. That is absolutely right. But I asked you what other stages it has to go through, okay, before it goes into G0 phase. Okay, so just think about it as we move further. Okay, and then in G1 phase, basically when we talk about the metabolic activity, we understood that the cell requires a tremendous amount of manufacturing to happen. So that is why it prepares a lot of enzymes, a lot of proteins, a lot of lipids, etc. And major goal for this cell, which undergoes cell division in the G1 phase, is to prepare for DNA replication. So, in order to undergo DNA replication, DNA replication is not a simple process; it's a very complex process. So, obviously, what the cells will do is the cells will try to manufacture and bring everything in order. For example, if you take the DNA replication as we studied, it requires a lot of proteins, okay, enzymes, okay, proteins and enzymes which are like not small in number, they're like very high in number. So these things have to be manufactured along with the availability of nucleotides, such as in the biomolecules you have studied that the DNA is made up of A, T, C, G, and along with that you need energy, okay. So all this has to be in a preparation before it goes into DNA replication, okay? And it prepares for that in the G1 phase, basically for DNA and DNA replication. And it also synthesizes sufficient amount of RNA and proteins, okay, to accomplish this process. Got it? And then we have our S phase. In the synthesis phase, we understood that synthesis phase is a phase where we have the process of DNA replication. So DNA replication means duplication of DNA. It's in a simple terms, guys. If you want to understand what is a synthesis phase, you just have to take a simple common word that you come across in your daily life that is called as photocopy. Okay, photocopy. So what are we doing? We are trying to make sure that if this is the parent DNA, this is the DNA in one cell. When it divides into two cells, it has to have the same equal amount of DNA. If it's a mitotic division, if it's the meiotic division, it should have half the number of DNA. For both the process, you need to make sure that your DNA is doubled, correct, right? So that is why the duplication of DNA will happen in the synthesis process, okay? But one key thing that we understood is if DNA and if you have chromosome, okay. So I told you in the last class already, if DNA is 2C and the chromosome is 2N, so tell me guys, by the end of the DNA synthesis, what will be the amount of DNA? What will be the amount of chromosome? Can you please answer in your chat box? I will repeat my question. We have the synthesis phase. After the accomplishment of synthesis phase, what will be the volume of my DNA, or what will be the amount of DNA after the synthesis of, after the synthesis phase, and what will be the amount of chromosome? Can you please try to answer this question, guys? Yeah, by then I'll just try to check on, yeah, somebody answered very quick. Very nice, guys, very nice. Okay, 2N, and 2N, okay, I will take it for chromosome. So one of your friends says that since I have mentioned as 2C and 2N, so he says that by the end of the, he or she, okay, I'm sorry guys, okay, I didn't really see the name, but anyways, by the end of the synthesis phase, you guys are saying that the chromosome will be 2N. What about the DNA? What will be the volume of DNA by the end of synthesis phase? Anybody? I think Rakshita answered. That's a correct answer, Rakshita. Okay, appreciate. Yeah. So you have the 4N. So it says that Trisha says that it is 4N. Absolutely right, guys. Okay. I mean, like 4C. I think that's what you meant. 4C. It should be 4C, guys. Remember, N is for chromosomes and C is for DNA. Don't try to mix up N and C. Okay. I know that it is quite confusing, sir. Chromosome is C, so it should be C. But why N? So always the DNA volume is given in alphabets as C. So you're right with your answer, but make sure that your alphabets are also right along with it. Okay, excellent. Okay, this was nice. Okay, so chalo. So you guys are right. By the end of the synthesis phase, the amount of the DNA that is present will be doubled, whereas the amount of your chromosome, okay, would remain the same. What happened? Okay, excuse me. I think, can you guys see the screen? I don't know. This folder. Yeah, I think you can see the screen. Sorry. Yes, yes. It turned blank, guys. Okay. Yes. So, and then we understood that this is what the synthesis phase will lead us to. Array, yaar. Okay, this is what the synthesis phase will lead us to. And excuse me. I can see only, yeah. All right. And then we went to the G2 phase. In the G2 phase, the growth 2 phase will happen. So the synthesis of DNA is already happened and the DNA has doubled. So now what will happen? Now the cell has to make sure it manufactures certain proteins and it brings back certain cell organelles also. For example, if the cell is trying to divide, okay, if the cell is there and if the cell has to divide into two daughter cells, obviously it's not just a, you just make the cell into half. It's not that easy. We understood that. So for that, what we need is we need certain powerful cell organelles like centromeres, okay, centrioles, sorry, centrioles, and then you need the proteins. For example, if you just look at the centrioles, yesterday we talked about centrioles in rough manner, but anyways, we understood that the centrioles are the cell organelles which are perpendicularly arranged, like here and here during the cell division. So these centrioles, what they do is they produce spindle fibers. I think you are a little bit aware because you've been taught in the NCERT class. But anyways, I'll be explaining about this in detail as we move further. So they produce something called as tubulin protein. Okay, they produce something called as tubulin protein. So all these things does not come easy. It comes with the preparation of the cell before going into the mitotic division, you see. So it has to make sure that these centrioles originate, they take their position, and we have the synthesis of a lot of RNA and proteins. Why proteins, sir? Why are we talking about proteins so much? So I've told you already that proteins undergo around 98% of all your biological functions. That means if the cell has to divide, if the cell has to signal, if the cell has to increase the speed of reaction, decrease the speed of reaction, and for any single aspect you need proteins, either in the form of enzymes or in the form of signaling agents or in the form of immune agents, something. It takes its role. So that's why the protein synthesis becomes a very, very important concept. Okay. So the cell grows in size, it increases its cytoplasmic volume, it synthesizes its DNA and also the protein along with it. And the cell prepares for something called as the M phase. The M phase is also called as the mitotic phase. Correct? No, guys, you agree with me? It's also called as the mitotic phase or the meiotic phase. So M phase is where the actual cell division takes place. That's what we discussed. So in M phase, what happens? Uh, you know, you can't really judge how long it takes for an M phase to complete or a cell division to complete in each cell. So it depends from organism to organism, tissue to tissue. So we can't really generalize. For an M phase to continue, it will take exactly the same amount. Okay. For example, if you take the human cell, it will take about an hour to complete this M phase. If you take an E. coli, for the complete cell division, it will take about 40 minutes to decrease. So it's a 20 minutes reduction in time. And if you take the human cells, it's not for all the cells that it will finish within an hour. Some cells take even longer. So we will generalize a human cell, a cell cycle. A general human cell cycle will take about an hour to finish, especially the M phase. Okay, all right. So M phase, we understood that this is the phase where the cell division takes place. All this time, what we studied is G1, S, G2 phase. All these phases are preparatory phases. That's why we put this phase as interphase. And that's why we call it as the resting phase. Here we don't observe, if you observe through the microscope, you don't really understand if the things are happening. You can't really observe through your eyes that if the things are physically changing. The most of the activities are metabolical in G1, S, and G2. But when it comes to M phase, in M phase, you can physically see the division of the nucleus, the division of the cell. Okay, you got my point, guys. So that is why the M phase is a very small phase in all of the cell cycle. So when we talk about M phase, we can divide the whole of cell division, we can generalize it M phase as cell division phase. In the cell division phase, we can divide it into two important phases. One is a karyokinesis, other one is called as cytokinesis. Karyon means nucleus, and kinesis means division. Cyton means cytoplasm. Kinesis means cytoplasmic division. Okay, guys. So what is happening here? Karyokinesis. Oh, all right. Now what is happening? Let's look at this in a different aspect. Now I have a cell. Let's say that this is my hair cell. This is my hair cell. So hair is quite negligible for certain group of humans, especially boys. We do care about the hairstyle, but we don't really bother much. But for the girls, hairs are very important. They grow a very long hair and they have a lot of patience for grooming. I understand that. But anyways, hair is quite, let's say that it is quite neglected in our whole body. So if you take the hair cell and if you look at the amount of the chromosome that is present, we know that it is 23 pairs of chromosome or it is 46 chromosomes even in the hair cell. In all of your body cells, it is always diploid number of chromosomes because you are a diplontic organism. Now, what I'm talking about is, guys, these 23 pairs of chromosome holds around 3.3 billion base pairs of DNA. What did I just say? What did I just say now? You know that your DNA is a double helical structure, guys. You know that it is a double helical structure. I know that you know this. If you don't know this, you can comment in the chat box. That's what are you talking about? I don't understand. How can I say that DNA is a double helical structure? That means DNA is made up of two chains. Okay. This is also one polynucleotide chain. This is also one polynucleotide chain. You would have seen the figures in all the science books that excites you. There is a picture like this of the DNA. So if you see the human DNA is made up of around 3.3 billion base pairs, or it is around 6.6 billion base pairs. Billion, guys. It is around 9 zeros, I assume. One beside 9 zeros. So these many base pairs. So what are base pairs? Let me tell you this. If you tell adenine, thymine, cytosine, guanine as your base pair, this is your base. And the pair for this, for example, if you have the DNA, we understood that if A, T, C, G is present, in the opposite side, it will be T, A, G, C. So these together will be the pair. You understand my point? These are called as what? Base pairs. This I discussed when we are talking about the structure of DNA, but I hope you understand. If you don't understand, you can ask me again, okay? So this one is called as a base pair. This one together, this one strand, one base, and the other strand, another base. This together is called as one base pair. Like that, how many we have? We have around 3.3 billion base pairs. And guess what? Even one single base pair goes missing. If one single base pair is replaced or it is not being added or something goes wrong, you will not be a normal human being. You will be a completely abnormal human being. You understand my point, guys? So that is why the cell cycle, especially in this M phase, karyokinesis is such significant process where all the nuclear components, that is your DNA, which is already duplicated, correct? No, your DNA is duplicated. This duplicated DNA has to be separated very, very carefully among the two cells or among the four cells that is going to come in the next generation. Are you following me, guys? Yeah? Yes, no? Can you confirm? Yes. Very quick, very quick. Thank you. Okay. So I hope, yes, I hope others are also understanding. If you are not understanding, guys, you feel free to just text your doubts. We can talk about it. Okay. So this is about karyokinesis. Karyokinesis is like the complete division of the chromosomes, which are duplicated. The DNA that is duplicated has to be repacked. I told you, like last time, when you're going to a trip, when you're going for a tour, you don't just pack your bags just randomly. You just pack very clearly and make sure that it is very systematic, and you fold your clothes and you put it in a bag and make sure that you carry the bag until you reach your destination. And what happens After you reach the destination is none of the concern, but you want to make sure that all your things are in place before you head there because you are uncertain. Similarly, in karyokinesis, a nuclear division happens, and nuclear division is not a simple word, it is a very, very complex process, and we are going to talk about it. Okay, and cytokinesis is the division of the cytoplasm. Okay, and some of the cells, we talked about some of the cells which do not divide any further, which undergoes the rest phase forever. Correct? No? We talked about it, guys. So if you talk about the rest phase, for example, if a cell is there and the cell decides that it will divide and it gives rise to two daughter cells. And two daughter cells will no more divide, no division. Okay, no division and no division. What are the phases that it has to undergo? Once this cell is born and it shall not divide any further, it will not divide any further, this cell has to grow itself in size and mature itself. For that, what it has to do, it has to go under G1 phase. The G1 phase of the non-dividing cell and the G1 phase of the dividing cell is completely different. In the G1 cell of the dividing cell, we understood that they synthesize proteins, RNA, and as well as they prepare for the DNA synthesis. Whereas the cells which do not divide any further, they will not have to prepare for the duplication of DNA. Instead, what they do is in the G1 phase, they increase their cytoplasmic volume, they increase their energy storage, they increase their organelle function, and then they stop. They involve in very specific function. Example, your nerve cells, your nephrons, your cardiac cells, your heart cells, etc. So once they finish the G1 phase, they directly jump into some other phase. It is called as G0 phase. And G0 phase is also called as the quiescent stage. So in most of your matured organs like your brain, your kidneys, okay, the nephrons in your kidneys, the heart cells, they do not go under any further cell division. You understand, my guys? So when they don't go under any further division, these guys, they finish their G1 stage and they directly jump into the G0 stage. As simple as that. Okay, I hope you understand. But they remain metabolically active, but they do not proliferate anymore. Okay. And now coming to the M phase, which is the mitotic phase or the meiotic phase, we can differentiate the cell division into two different important, significant types of division. Okay. One is the M phase can be divided into two different types of cell divisions. One is mitotic, other one is called as meiotic. So in mitotic division, it is called as an equational division. So equational division means if you have a cell and if you have a nucleus and the nucleus has the chromosomes, so if it is in 2n in number, when it divides, the two daughter cells that come across will always have the equal number of chromosomes. So you can ask me the question, sir, is mitotic significant or meiotic significant? But let's come to the general basics of this. In mitotic, it is called as an equational division. When the equational division happens, you have a mother cell, which is her chromosome number is 2n. Let's take that our human beings. Some people will understand 23 pairs of chromosome, and some of you guys understand that it is 46 chromosomes. Both of you are right. If you say that it is 23 pairs, you are right. If you say that it is 46 chromosomes, you are also right. So now this mother will divide into two daughter cells. Now, by the end of the cell division, if the daughters have both of them have 46, 46 or 23 pair, 23 pair, it is called as a mitotic division. What about the meiotic division? In the meiotic division, what happens is the mother cell is diploid. The mother cell will divide into, by the end of the cell division, she will give rise to 4 daughter cells. And all of them will be having half the number of chromosomes. You understand, guys? So mother was 2n, but the daughter cells are n. So to understand the significance of mitosis and significance of meiosis, let's not rush here. We will try to make sure that we study one step at a time. Our focus, let's be on one type of cell division, that is our mitotic division. So mitosis Mitosis is also called as equational division. So before I go into the details of equational division, let me talk you about where this particular cell division occurs. Okay. Now let's say, guys, you are playing in a playground and you just fall down and you have a cut. Now you have a cut. You might be bleeding, or your just skin cells are just just rubbed off. Okay, the grading, like how you grade vegetables, it's just happened like that, or you had a deep fall where you need the stitches. But anyways, you have a cut. When you have a cut in your body, the bleeding might stop. That is a different subject. But there is a gap, no? Your skin cells has a gap. This gap. Who fills this gap? There are some tissues that are damaged. There are like thousands of cells that are missing. Who fills this gap, or how it fills this gap? So this repair mechanism is brought about by your vegetative cells. I told you that your vegetative cells are also called as somatic cells. Your somatic cells are responsible for repair, growth, regeneration, multiplication. My handwriting is very bad, guys. I'm really sorry, but I hope you can understand what I'm writing. Okay, if you don't, if not, please try to put it in the chat box. So I will try to make sure I write slower. Okay. Repair whenever you have a damage. Sir, I don't have any damage. Does it happen in a normal time? Guys, remember, by the time you wake up and by the time you sleep, your body has gone into a lot of damage. So that's why you sleep, and that's why the sleep is very important. And at this point, all the cells that are almost old, that are not functional, they have to die, and these dead cells have to be replaced, like your skin, like some of the organelles, like liver cells. They go under replenishment. You just become young. You shed your skin like around 1,000 times within the time, by the time you die. From the birth to death, you almost change your skin. The whole skin will change for thousands of times, 1,000 times. That's when you need repair, and replacement is required. And then it is a growth. So you know that you started from the single cell and you have to go through till the adulthood, and then you age and you die. So, but you have to start from a single cell, microscopic structure. You are a microscopic structure. From then you become this 30 trillion. Somebody said 37 trillion. I don't forget. That is average of 37 trillion cells you have to become. For that, what you need? You need a division. Next comes regeneration. Like your liver is a regenerating organ. It goes under damage. So these cells have to be replaced exactly like how it is. And then multiplication. Some of the cells are very few in number, like your sperm mother cells or ovum mother cells. They're very few in number when you're born. But as you grow up, they have to multiply. These mother cells have to multiply into several thousands, so that to support your reproductive process. An example I'm telling. So this multiplication also happen with respect to mitosis. So what is the significance of mitosis? The significance of mitosis is it does not it makes sure that the volume of the genetic content, that is the chromosome number, always remains the same in each division, whether it is repair, whether it is growth, whether it is regeneration, whether it is multiplication. Clear, guys? So the significance of mitosis is tremendous. That's what is it's enabling you guys to repair and to regenerate, to feel young. If you work out, if you want muscles and anything, whatever you're just doing in your daily process, if it has to be accomplished, you need your mitosis. Okay, so I hope you understand the significance of mitosis. Now, coming to the mitosis, we understood that it happens in somatic cells, and somatic cells are also called as vegetative cells, which are all the cells in your body except germ cells. Okay, germ cell. What are our germ cells? Germ cells are your sperm and ova. Except for that, all of them undergo, all of them are called as what? Somatic cells. Okay, and it is also called as equational division. Done. Let's go, guys. So this mitosis or the karyokinesis, especially, you have two phases. No, mitosis is a cell division. It's a type of cell division. It can be divided into two stages. One is a karyokinesis, other one is a cytokinesis. Correct? No? So under karyokinesis, we can divide the mitosis into four different stages. That is the prophase, the metaphase, the anaphase, and the telophase. I think you guys are aware of what are these names. Are you aware? Have you heard about these guys? Have you studied in your school? Yes? Yes, no? Come on, I've been talking. Hold on, let me not get confused with the time. Okay. Have you guys studied about mitosis? Have you studied about these phases in mitosis? Which talk about prophase, metaphase, anaphase, and telophase. Have you heard about it? No. Very good. It's very good that you told, because that enables me to go in little slowly and little detailedly. Okay, so I will make sure that you understand. Okay, all right, guys. So if some of you have heard in your, oh yeah, even Trisha and Rakshita, I don't know, other guys, come on, guys, participate. Sanjana, where are you? And then we have the guys from LD Valley, and then we have Sugong. I'm sorry if I pronounce your name wrong, because it happens, guys, you know. So if I'm wrong with the pronunciation, please forgive me. So other guys, I want you to participate as well, because I want to get to know you better. So anyways, guys, I told you that we can divide the whole mitosis or any cell division into cytokinesis and karyokinesis. And karyokinesis is first thing that happens, and the cytokinesis is the second thing that happens. Even in mitosis, the karyokinesis is the first thing. Here, what's happening, guys, is you can divide the whole karyokinesis into four phases, four important phases. That is the prophase, the metaphase, the anaphase, and the telophase. Okay. So if you don't remember the words and the names of it, chill. Don't worry about it, because as I describe these phases, you will understand what's really happening. Okay. Now, the first phase is the prophase. Okay. See, come back here. You have to remember the order of the events: prophase, metaphase, anaphase, and telophase. After prophase comes meta, and after meta comes ana, and after ana comes telo. So you have to remember the sequence of these stages. You can't mess this up. And guess what? This is just the beginning. In the meiosis, it gets even more complex. That's why I want you guys to feel easy here, so that you take the same way even in meiosis. Okay. So let's go for prophases. So among all the four phases that we see here, the prophase is the longest phase. So what's really happening here, guys? See, if you just try to summarize what we just studied, we studied that the cell undergoes G1, the cell undergoes S phase, the cell undergoes G2 phase. By the time it has come to G2 phase, we know that in the synthesis phase, the DNA is doubled. Okay, I will write it as 4C. The DNA has doubled. Now, what is the state of my cell? My state of cells look somewhat like this. This is my cell, which is almost ready for karyokinesis or cell division, and it has an intact nucleus. Intact means a very proper nucleus. Within the nucleus, all the genetic material, okay? What is our genetic material, guys? It is DNA, okay? And how is our genetic material packed, guys? It is packed as chromosome. Agree? If you have any doubt with these two things, please ask me. So the DNA is our genetic material, which is found in the nucleus. So it is so much big, okay? And these things have to be packed, and they are packed as chromosomes. So if you don't know what is chromosomes, they are like X-like bodies, X-like structures, which are found in your nucleus. Got it? So now, by the time the cell has finished the G2 phase, you don't see any chromosomes. What do you see? You see that a thread-like structures just scattered within the nuclear regions, a tremendous network of thread-like structures, a random thread-like structures just floating in the nuclear region of your cell. So this is the beginning. This is called as pre-prophase. Pre means before prophase. What is my stage of the cell? So my cells were metabolically active. No, if they are metabolically active, What are the important cell organelles? Can you just summarize? My cell requires mitochondria, correct? No? What is mitochondria? I think you guys are aware of mitochondria more than anybody else. If I ask you what is mitochondria, you guys will tell that mitochondria is a powerhouse of the cell. Agree with me? So what does mitochondria produce? ATP. So anyways, it is the powerhouse of the cell. Without mitochondria, most of the functions do not take place. Of course, if you don't have the generator, if you don't have the UPS, if you don't have the power supply, you don't do any functions at home. That's what is happening here. So mitochondria should be there in the cell, correct? And next one, what do you have? You have Golgi apparatus. Golgi apparatus. Okay. What is Golgi required? Golgi does so much functions. You take the protein, protein manufacturing takes place somewhere. It takes that manufactured product. It will modify this product into functional products. It will pack. It will send to a certain destination. This guy has a lot of functions. Who? Golgi apparatus. You should have studied this in your cell, the unit of life. If you don't, if you still have doubts, by the end of the class, you can tell, sir, from the cell, the unit of life, these particular topics I have doubt, we can talk about those cell organelles. Okay. See, this Golgi apparatus has a very specific function. So they make a lot of roles. Okay. So let's say packing and moving. Packing and moving. Okay. And what is your cell have? What is this? This is called as nuclear membrane. Nuclear membrane. What is this called? This is called as cell membrane. Sorry, guys, I'm taking the lower space. If you can't see in your projectors, please let me know. So this is called a cell membrane. See, before the prophase, the cell has all the cell organelles like the mitochondria, like the chloroplast, like the Golgi apparatus, like the nuclear membrane, cell wall, etc. The cell is very, very normal. But now what happens is the cell is entering into the state of division. That means it has to divide. The first state of division, now pre-prophase, I told you, these were all there, guys. These were all there. Now the cell is ready for the cell division. What are the changes that we can see in the cell that is going under cell division? Now I told you that your cell has the nucleus, and the nucleus has a genetic material, and the genetic material is so randomly, just like threads. Guys, just remember, if I start removing each and every chromosome, and then if I place one nucleotide, one base pair with another base pair with another, it will be around 2.2 meters in length. That is the length of the DNA in every cell that you have. You can't even see the cell, but 2.2 meters of DNA is just present in each nucleus. You understand? So much of randomly just wires, just like spread away. Now, what is the role of the cell? This DNA cannot be distributed. Like if you are going for a trip, you can't just go pack the whole wardrobe. You just take the wardrobe, dump in your bag. Can you carry it like that? No, your bag has a limited space. You need to make sure what is required, what is not required. And you need to make sure that everything is in order. Similarly, what happens? This genetic material that is so loose, which is completely untangled, they go under a process called as condensation. What is condensation? Condensation is a process of packing of DNA. The DNA, which is just like threads, they use specialized proteins where they wind this DNA. For example, this is your DNA. They use, see, guys, let me make sure. Your DNA is negatively charged. These proteins are positively charged. What happens to the opposite charges? They attract with each other. Similarly, our DNA uses simple physics. And here, the DNA is negatively charged. The proteins that are used for packing is positively charged. Now, the positive and negative attract with each other. Now, the process of winding, process of packing takes place. So, this process is called as condensation. So, the DNA which was like thread-like will become what? Will become nucleosomes. From nucleosomes, what they become? They become highly condensed thread-like structures called as chromatin fibers. From chromatin fibers, what they become? They become very, very structurally organized structures called as egg-shaped bodies called as chromosomes. You already end up like this. From thread-shaped to nucleosome, from nucleosome to chromatin fibers, from chromatin fibers to chromosome. So this process is called as the condensation process. So first, the nuclear condensation happens. What is nuclear condensation? The packing of DNA occurs. Okay, are you able to hear me well, guys? Are you following me? Yes, no? Can you confirm, please? Yeah. Perfect. Thank you guys for the response. So now we have the chromosome. The nuclear condensation has happened. And now from the DNA, just the random threads, they have gained a specific shape. And our specific shape is getting our chromosome. The chromosome is being condensed. You see that from thread shape to chromosomes, this is happening here. Now, simultaneously, I'm not telling that this one step after another, as this process is happening, simultaneously, some other process is also happening. What is that one important process? One important, like there are many important processes, but here, what happens is the major aim for your genetic material is to be separated, correct? No, your DNA volume is doubled, or the double DNA has to be separated between the two cells. Who does the role of separation? The role of separation is coming from your centrosomes. The centrosomes are your cell organelles, which are like perpendicular structures. They, what they do is they would have already appeared, they would have already formed and taken them. They are not present all the time in the cell. They're only, they come into place only when there is a need for cell division. So when there is a need for cell division, these centrosomes, what they do is they come and occupy the pole positions. What are poles? The south pole and the north pole. Similarly, the upper pole and the lower pole of your cells. They come and occupy. So these guys are in place. They are preparing for something. Along with them, what will happen is you have your nuclear membrane. So this is your intact nuclear membrane. Intact means very, very properly formed nuclear membrane. As the prophase is happening, this nuclear membrane starts disintegrating. When I tell disintegrating, it starts disappearing. The nuclear membrane starts becoming lighter and lighter and lighter and finally disappears. You know what I'm saying? So why the nuclear membrane has to disappear? Guys, you have a gate. You have a gate for your home for a particular reason, for security reasons. And when you have a function, when you have an event, will you keep that gates closed? You make the modifications for your gate so that more people can come easily without having the security checkup. Correct? No, they can come, they can enjoy your home, they can enjoy your hospitality, they can do their job and get lost. So that is what is happening. So similarly, here also the nuclear membrane is very, very strict. They do not allow any other things to enter into the nuclear space. That's why they have to disappear before the separation happens. So the disintegration of the nuclear membrane takes place. Got it now? Got it? So what did we study? First thing, we studied that the genetic material, which are like the random threads, the DNA that is scattered is packed and condensed. They're packed into chromatin fibers and chromatin fibers into chromosomes. Next, what is happening? We understood that centrosomes are a very vital part of separation of these chromosomes. So they take the polar positions, they take the opposite positions. And third thing, for us to make sure that we separate this genetic material, we cannot enter with the presence of nuclear membrane. So that is why the disintegration of nuclear membrane takes place. So the nuclear membrane disappears slowly. And is that it? Not really. Along with that, you have your some of the core mitochondria, Golgi apparatus, or Golgi body, they both disappear. During what phase? The prophase. Remember this. The Golgi apparatus and the mitochondria, they disappear during the beginning of the prophase. Okay? So these are the events that happens in the pre-prophase and the prophase. Guys, do you guys understand? So what can we see by the end of the by the end of the prophase? What can we see? By the end of the prophase, what you can see is your cell is like this. There is no mitochondria. There is no Golgi apparatus. The nuclear membrane is barely visible, but the chromosomes are quite condensed. You know, the DNA material is quite condensed, and the centromere has taken its place. Now the centrosomes have taken its place. The centrosomes, what they do, you know? What is the role of centrosomes? The centrosomes have to produce threads. You know, guys, for example, when you do fishing, when you want to fish without a net, what do you use? You use a fishing pole, and you know there is a fishing hook, and then you just put the fishing rod like this, and along with the fishing line, the hook goes and falls in the ocean or river with the bait. Correct? No, the same mechanism happens. Imagine that this is your fishing pole. This is your fishing pole. Who? The centrosomes. We have to talk about centrosomes if you have to understand. The centrosomes are like this. What they produce is the tubulin proteins. The tubulin is a type of a protein, and these tubulin proteins, when they are formed completely, they are called as spindle fibers. They are called as spindle fibers. So these spindles, what they do is they're highly specific, guys. So you see these chromosome structures. This is how, sorry, I will write it somewhere here so that it's not sloppy. So let's say that this is your chromosome, and this is where the sister chromatids are attached. So this is called a centromere. Centromere. So here what happens is you have a disc-like structures on the face of the centromere, and these are nothing but kinetochores. So what are kinetochores? Kinetochores are like hooks. They're like the point of attachments. So they're very, very specialized to understand this is a spindle fiber. So when a spindle fiber is formed, they get hooked on like a fish. Like they get hooked on to this kinetochores. Now, your centrosomes, they are like powerful motors, and we call them protein motors. They are called as what? Protein motors. So what are motors do? They spin. When they spin, what will happen, guys? They start winding and winding and winding. When they wind, what are they doing? They are fishing your, you know, sister chromatids. They are pulling your chromosomes away. So this is pulled away. This is also pulled away. You know what I'm saying? In opposite direction, they separate. So you understand what is happening. The spindle fibers are formed by the centrosomes, and the centrosomes form the spindle fibers with the help of a protein called as tubulin. So tubulin protein is formed, and the tubulin forms these powerful spindle fibers, and the spindle fibers attach to the center called as a kinetochore. And once they attach to the kinetochore, once it is hooked, once it is in place, now they have a signal. Okay, I have hooked on to this particular chromosome. Now, what's my job? I have to pull these chromosomes away from each other. Why are they pulling? Because the number of chromosomes or the number of DNA has to be equally distributed. We have double DNA, no? So this double DNA has to be equally distributed among the daughter cells. So that's what is happening. Are you guys clear? What's our time? Okay, we're almost at the end, guys. So all these things, I know I can push it faster, but this is quite necessary for us to understand. Is it clear? Did you understand this concept, or am I too fast? Guys, I hope you're here. Awake, sleep. Right. Other guys, you can just put it in the chat box, or you can, oh, okay, this cell is not dividing what stage. That's me. Hold on. What's the difference between a kinetochore and a centromere? Okay. Okay, let's say that we have, see, a chromosome is made up of, see, let me give you, this is a very important question, very beautiful question, because if you don't understand this, it's going to create problems. Okay, I hope you guys understand. Okay, guys, listen to me. Do we have 23 pairs of chromosomes? You agree with me? Yes. So let's say that I put this chromosome as like this, like this, like this. You might call me mad, but what I'm doing is this is 1, 1, 2, 2, 3, 3, all the way it goes to 26, 26. Agree with me? 23, 23. Sorry, guys. All the way it goes to 23 and 23. What am I saying? For every one, there is another one. This is called as chromatic pairs or chromosomal pairs. You understand my point, guys? So if I tell 23 pairs of chromosomes, total there has to be 46 chromosomes. So for one, there is another one. For two, there is another two. So if I talk about one, this is one, this is one. So what do I call these chromosomes which are belonging to the same category? So they are one. No, one is not same as two. Agree with me? So these one or two, they are called as what? Homologous chromosomes. Okay, they are called as what? Homologous chromosomes. Okay, so each pair, this is a homologous to this. So if one is there, I will call it as homologous pair. Got it? No, this is one term. You remember this is called as homologous chromosomes. Now, I take this one. Hold on, guys. I don't have space. Okay, let me erase this. So I have this chromosome. I had one. This is one. I call it as homologous pair. Now, what do I do is I take this one. If I take this one, what I see is this is made up of two sister chromatids. Two thread-like structures have come together to form one chromosome. So these two thread-like structures are attached at the center point, and that center point is called as a centromere. This is called as a centromere. A centromere determines where the two sister chromatids are attached. So if you see your cell, the unit of life, based on the position of your centromere, for example, if the centromere is here, if the centromere is here, if the centromere is here at the far end, if the centromere here at the last end, we can call it as metacentric, submetacentric, acrocentric, telocentric. You can see this in your textbook when you see the cell, the unit of life. So the centromere determines where these two sister chromatids come and attach at a point. Now, what is a kinetochore? Kinetochore is something that is present on the centromere like a disc-like structure. Now, what is the role of this kinetochore? This kinetochore acts like a point of attachment to the spindle fibers. So once the spindle fibers comes and attach to this kinetochores, now what will happen is this will hook, and the centrosomes, what they will do is they will start spinning their powerful protein motors to separate the sister chromatids. You see this? This is the chromosome. Either they take the whole chromosome away, or see here in the mitosis, what will happen? Both the sister chromatids are dragged to the opposite poles. But in meiosis, the sister chromatids are separated like this, one and one, like this, something like this. Okay. So the difference between a kinetochore and a centromere is that the kinetochore is a point of attachment for the spindle fibers, whereas the centromere is a point of attachment between the two sister chromatids. Is it clear, Rakshita Telwar? And I hope this question helps you guys, other guys also, because it's a very, very pretty important question, very excellent question. Because if you don't understand what is homologous chromosomes, what are sister chromatids, okay, I would have explained you as we move further. But anyways, this came at the right time, guys, very right time. I appreciate the question. Okay, so that's it. Okay, hold on. So others, I hope it's all clear. So I'm running out of time, guys. So we have to end the session here. But the next session, we're going to push even faster. I keep promising you this, but if the cells is not dividing, what stages it undergoes? Is it me? Oh, this is a very early message. Yes. Okay. You guys have told yes. This is quite messy, guys. Yeah. Cool. All right, guys. Thank you very much for attending the session. Before you go, as I told you, there is always an assignment for you. This is like a help you're doing me by the end of the session. Please attend the poll so that we can improve our classes. And the people who feel it's very slow, please adjust, because not everybody is in the same phase of learning. I'll make sure that the important points are reaching you at the right point. Okay. Basics are very, very important to understand any complex topic. Okay. Oh my God, we have so many messages. Yeah, Sakshi Kumari, thank you so much. Yeah, I just saw your messages in the YouTube. Okay. Yes, exactly, exactly. So whatever you have given the answers, most of the astral rays or the astra projections, yeah, packing up DNA, one cell is equal to two astra. Very good, very good. Sakshi Kumari and Aditya Singh. I'm sorry, guys. I just didn't give the attention much to the YouTube. I hope that I will just look at it in the next class and I will communicate with you as well. But your answers are very, very clear and very, very deep. I appreciate your communication, even though it's in YouTube. Uh, thank you, guys. Yeah. Thank you, guys. Thank you for giving the poll and your feedback. I'll see you in the next class. Have a nice day. Ciao, ciao.